Okay, so thank you, Jacob. Thank you for the invitation to talk to this group today. I'd like to give you a brief overview of CIFR and then introduce to you, which is probably not new to you, big data, what it means, with a particular focus on microbes and microbial diversity and what CIFR is doing to tackle this and where it's going to lead and what it's going to do to help agriculture, to manage disease, increase plant yields. So first of all, I need to introduce to you the members in our center because this is very unique. The center was structured so that we have expertise that covers all components of an ecosystem. So if you're thinking about soil ecology, we have experts, we have experts that focus on that, epidemiology, fungi and bacteria, which are the two major players, population genetics, molecular pathogenesis, also secondary metabolites and fungi, and more broadly, another initiative started up by Gary Payne is the Amplify. We also have expertise in soil biogeochemical processes and plant ecology. So we're basically covering both above ground, below ground, abiotic and biotic processes that you would see in ecosystems. These are members from CALS, the College of Agriculture and Life Science, but we also have members from the College of Science who are experts in statistical genetics, mathematical modeling, and climatology. This is critical because ultimately what we want from these microbial diversity studies, from these big data studies, we want to model systems. We want to make predictions as to what will happen when there's a perturbation to the system, be it a chemical, be it environment, climate. How does the system adjust? What shifts do we see in the microbial communities? If we can understand the process and it's recurrent, we could use that to our advantage. So that's the real goal here, is to understand process. It's not enough to know pattern. You need to know how microbes are changing through time and space. And this is our mission. We are dedicated to understanding this, and we want to understand it at various genetic scales. For example, we want to look at within cells, spores, and I would point you to this vignette here with Aspergillus, these chains of conidia, because we know that within cells there's extra chromosomal elements, supernumerary chromosomes that play a role in what these microbes do. But then we want to go up to the individuals and the population scale and know how genetic material is transferred between individuals, because ultimately we need to track the individual. A single locus is not a marker that tells us what the individual is. These organisms are known to undergo frequent genetic exchange and recombination. And what's captured, this is captured best in a phylogeny. Think about a phylogeny where you're looking at lineages of organisms through time and space. And the idea is ultimately you want to connect that to the biopesticides that you develop. Once you have this understanding of the process, this becomes even stronger, the development of these pesticides and knowing that how they change through time and how this will inform which ones you introduce into the system and make predictions as to how the system will adjust given that the environment changes. So clearly this is a multidisciplinary effort. It requires experts in the biology of these organisms, but also in the computation, the genomics, and we're dedicated to the training on these methods, sharing the resources. Wouldn't it be wonderful if you had spatial maps of microbial diversity and you had this on a scale that's beyond species, but on a population scale? You could actually type individuals, just like they do in criminal databases. This is ultimately the goal. And we want to share this with the community. These resources should be available. These databases grow on a yearly basis. This should be a resource that's made available and ultimately engage stakeholders so that there's more funding in this area of research. Okay, so you're all familiar with big data. The big thing here is high-resolution digital, and you've heard of the drones, the UAVs flying over these fields and getting digital maps of soil moisture, pest damage. And so you could actually, that's in a simplest scale. In a more complex scale, you have these three-dimensional images of the soil, root, and plant interface. And this generates a lot of data depending on the time scale. You could collect this data over a week, over an hour even, over months. And this generates a tremendous number of data points. Couple that with environmental parameters, timing and application of chemicals, fertilizers. You could see how this is an overwhelming amount of data. But one thing that hasn't been fully integrated into this is the microbial diversity. This is what we're all aiming to do. And up to now, we've been looking at abundance and composition, which is extremely valuable. It's a starting point to know what you have out there. And this has been possible with the advancements of sequencing, as you know. We could target these barcode regions, the internal transcribed spacer in fungi, or the rDNA, the 16S ribosomal RNA gene in bacteria. That's genus resolution. We get species resolution. That's great. We have a wonderful database thanks to the Earth Microbiome Project. We could blast our sequences and have an idea of who the players are. But it's not sufficient. And you've all been grappling with this. Those of you working in metagenomics, we can't just stop at these. These barcodes were developed more than 10 years ago. And we need to take advantage of the costs of DNA sequencing. And because the cost of DNA sequencing is plummeting, it's really outpacing what Moore's law, if you're familiar with computational power doubling every year. For the most part, the sequencing costs keep track with Moore's law, you know, in the Sanger days, but then once high throughput methods came, it's become very cheap to sequence. So to put this in perspective for you, a typical bacterium of 2 megabases, you could sequence 5 of them on the buck. Filamentous fungi, a 40 megabase genome, use genome reduction methods, bring it down to the size of a bacterium. There you go. You have a snapshot of the entire genome. You could look at deletions, aneuploidy, for the cost of what it takes to sequence a bacterial genome. We need to do this. Now, how do we use this big data? How do we leverage all this information coming to us? Clearly, we have real-time management decisions. We could reduce the amount of pesticides that we use. We could use irrigation more effectively. But one thing that we need to focus on is how do we use microbes to our advantage? How do we understand how microbes interact in the ecosystem? Clearly, you all know there's competition when you introduce multiple microbes. There's fitness costs. If we can understand this, and again, if we could type the individuals, then we have a way of enhancing our disease management methods, going beyond chemical protectants, host resistance, which has been the focus up to now. We can complement this with microbes. And then there's another emerging area. You probably are familiar, you've heard the toxicological reports. A lot of these filamentous fungi produce secondary metabolites that are toxic to humans and animals. And this is only now becoming apparent because there were no regulations for these. Think about the ochratoxins, the fumonisins. These are not even regulated on some crops. And these have debilitating effects not only on animals but humans. There's been toxicological reports of stunted growth in children that over long exposure to aflatoxins. So what will the microbial diversity? It will help us detect these particular strains, these species of fungi that produce these toxins. We could target them. We could then, the goal is to eventually understand how we could shift these populations to becoming non-toxigenic. And there actually are examples where this has been done. So this is the long-term vision. Now, there's challenges in doing this. And one thing is, with all this data, you want to model this data, you have data through time and space, and you're measuring all these components. This complicates the system and complicates the models. And in particular, we're limited with the microbial diversity data. And what's limiting about the microbial diversity data, in any given study, you search your nice genome, your CHIME, you do your search and you have your barcodes. Some of them don't match anything in the database, and others, they're too divergent. And what do you do in that case? Well, your program drops those sequences. So you're missing a lot of the diversity that's out there. And keep in mind, from an evolutionary perspective, the rare individuals often play a more important role than the more frequent ones. And we're just throwing out the rare. We don't find them. They don't fit in the database. It's great for a first pass and understanding what you have, but you need to do more. You need to drill down more, ultimately, if you want to know the process. And then how do we integrate all this information? Let's say we do know the players. Let's say we understand how they interact with other organisms. How do we then provide advice to farmers as the best strategy moving forward? Obviously, biopesticides that emerge from these studies are not very fast in their action. You know that. We know that we need chemicals. But that fraction where chemicals fail and we know that the microbes are playing a role, that's that we could fill in that void if we understand more about the microbial diversity on a finer scale than species or genera. So our focus is to really drill down and understand microbial diversity. And our specific angle here is that to really effectively do this, you need to understand how diversity changes through time and space. Microbes are not static organisms that you can barcode a single locus. These are dynamic. They undergo genetic exchange, recombination. Why is it that we can't sample a biocontrol strain after application after a year? Where does it go? Is there genetic exchange? What happens when we find that a pathogen is not a problem, that then it reemerges? Why is it reemerging? Where are the reservoirs? We are missing this information. So we need to improve our genotyping methods, and we can do this now. It's to add to the existing efforts, but to drill down and get to linking patterns of diversity to process and how that changes. Once we understand that and it's recurrent, we could use it to our advantage. The first three parts here, assessing microbial diversity. Yes, if we could do this, then we could understand clearly what are these microbes, and more important, the lineages or the individuals that confer these beneficial effects on plant health. Biomarkers, this word is used very loosely, but what is the biomarker that you're using? And is it effective to use one or two loci to really get a handle on which species are found predominantly in fertile soils or lineages? The interactions are extremely important, but again, that comes down to the players and how these are changing through time. And this is the recurrent theme that I'm putting out there, is that we really can't do this with a one-time sampling. This is a continued effort of looking at these populations, these communities through time, and to then enter all this information in databases that we can search to understand how environment impacts their distribution and how they shift. So ultimately, we want to model this. Okay? We want to have models that predict how a system will shift in response to a perturbation, in response to environmental change. Simple, just like pH varies along a gradient. Bacterial populations also show that variation. Fungi, there's latitude gradients. Why do we have these latitude gradients in diversity? But what specific lineages do we have there? It's easy for bacteria because their genomes are small, and you could pinpoint this. But in fungi, we are dealing with cryptic lineages. We don't even know the boundaries of what an individual or a species is anymore, because a lot of this is cryptic. You don't see it. They do undergo genetic exchange and recombination. So we need to understand better the boundaries of the genetic continuum in these individuals, these species, before we can actually develop effective long-term biocontrols or biopesticides. And that's where it comes with these ideas of sentinel species to understand their distribution. We know these latitude gradients exist. Perhaps there are species, individuals that are diagnostic of when the environment changes, you see this particular lineage increase or you see this lineage go down. So ultimately, the strength is to have the information, to share it so that it becomes one large effort, just like the Earth Microbiome Project, but on another scale. And this is the direction that we're moving in. And then to integrate it with other information that you have with about the ecosystem. Okay, and I think I'll just stop there. Thank you. There's no questions, right?